Hello, so first of all, I would like to thank the organizer for giving me the opportunity to present some of the work that we've been doing in my group in Novartis Oncology Data Science on developing a fully unsupervised deep mode of action learning method for phenotyping high-content cellular images. So I know that most people in the audience are probably very well familiar with high-content imaging, but I wanted to use this vignette so that we're all on the same page on how high-content imaging is used for mode of action identification and discovery. So you typically start with a cellular system, in our case, typically a cancer cell line, on which you apply a bunch of chemical or genetic perturbations before you image the cells with, say, fluorescent microscopy to obtain phenotypic images. And then two main types of pipelines coexist in the literature. One type that I would call the conventional pipelines, where you start with an object detection step, where you segment the cells from the image, and then you apply a bunch of predefined measurements on your cells, measurements that are typically like measuring the total intensity of a given channel, or the shape or the size of the nucleus of the cells, these kind of things. And using those pipelines, you convert images into a matrix of numerical descriptors that you use for downstream analysis. And then at the bottom of this slide, you see the deep learning pipeline, where the goal is to train a deep neural network in some way that then allows you to do mode of action identification and discovery. And for that, the question is really, how do we go from pixel to mode of actions? And so today I want to start by describing actually a supervised learning method for mode of action prediction from raw pixel values that is quite central to our methods. And that has the advantage of circumventing the need for object detection. But then I want to spend a couple minutes on talking about inherent limitations of supervised learning for mode of action prediction. And then I'll switch to describing our methods in full detail, which is a fully unsupervised approach for that is based on a deep learning clustering backend for feature extraction, and that very importantly incorporates state-of-the-art batch correction algorithms during its training procedure. And I'll finish the presentation by showing how we use this method for phenotyping unlabeled images. So all the results that I'm presenting today come from the well-known BBBC2021 public dataset that is from breast cancer cells that are treated with a variety of drug and image after 24-hour exposure over 3 channels that are DNA, tubulin, and actin. The dataset consists of above 13,000 images from 901 treatments. And perhaps best known is the annotated subset of this dataset, which consists of just below 4,000 images from 103 treatments taken from 38 compounds that are annotated with 12 distinct MOAs. So in the literature, lots of methods focus on this annotated subset because you have ground truth label on which you can train your supervised methods. And also it's easier to evaluate your approach because of the ground truth labels. So first of all, let me briefly describe the supervised approach that was developed by William Godinez when he was a postdoc in Novartis. So the method starts directly from the raw pixel intensity values of images without the need for an object detection steps. And it does so by working on the images at different scales. So on this particular cartoon at 7 different scales. And on each of these scales apply convolutional layers independently from each other before concatenating them, concatenating embeddings obtained from each of the scales into a single embeddings, and then applying a traditional classification output layer. The method is called MSNet for a multi-scale neural network. And typically you can predict mode of action if you want to focus on the annotated subset of the BBBC 021. or you can, something very smart that William did in his paper is to train the method on predicting treatments. So treatment is typically a compound and a dose because the exposure is constant here in this data set. And in this way, you can train your data set, you can train your method on the entire data set because the treatment information is always available. And really, the method is able to circumvent the object detection step because of this multi-scale process where it's able to detect both population scale effects by looking at very zoomed out field of views, and as well take into account local descriptors of cells by looking at very zoomed in version of the images. The method was published in 2017 already, and at the time it achieved state-of-the-art accuracy on the annotated subset, as you can see on the left. It actually gets a perfect accuracy for 9 out of 12 of the annotated mode of functions, and does a decent job at 2 others, and perhaps only for protein degradation could performance be improved. But also a very interesting output of the method, because it's a supervised learning method. Basically, the output is a probability for each image to belong in one of those 12 mode of actions. And so you can track at different concentration what this probability looks like. And here, this is what you have. So you have the mode of action probability on the y-axis and the concentration on the x-axis. And for compounds, like here on the top left, you can see that they're consistently over the entire dose range predicted as DNA replication. But for other compounds, such as this one, you can see that they start by being pretty inert, where actually the probability of DMSO is higher, and then switch to what they're annotated as, like kinase inhibitors. But then for very high dose, they actually behave more like a DNA damage agent. And this is a point that is very important, and we're going to come to that again a bit later. So despite its great performances, I think there is mainly 2 types of limitations of supervised learning for mode of action discovery. Perhaps the most obvious one is that we're limited from prior knowledge, right? We're limited to annotated data sets to train on. Annotations are expensive and rate limiting to obtain. And even if you train on treatment, so apply some kind of pseudo-supervised approach, then you're still facing other limitations. But the annotations also may be wrong, and just simply it limits our ability to discover new biology because we try to predict things we already know. But the second limitation, perhaps more important in my eyes, at least to what we're trying to do in my group, is that all target labels really are treated equally by supervised learning. It doesn't at all as a notion that some labels are very closely related to each other. For instance, similar doses of the same compound would be 2 treatments that would be treated as dissimilar as 2 treatments from compounds belonging to completely different mode of actions. And if you're work in a lead optimization setting, typically you want to compare closely related compounds at multiple dose and time points. And so supervised learning might bias you here. So the question we asked was, can we get away from using labels at all during training? And instead, we want to switch to an unsupervised deep learning analysis where you would start by training a deep neural network in a way that I will specify, but without using any kind of labels. And then at inference time, you would feed a new image through the deep neural network to obtain a feature vector, in this case 64 features in this vignette. And then you can use this feature vector to either visualize your compounds using t-SNE or UMAP or PCA, or to do a clustering analysis that can then lead you to identify mode of actions. And here the assumption behind unsupervised techniques is that similar treatments will cluster together because they have similar phenotypic effect on the cells, and therefore the images will look similar. So the procedure we use for unsupervised learning is based on a recent publication from the Facebook AI research group that's called Deep Cluster. It's a clustering-based feature extraction method that takes a neural network from which you remove the last layer if it's a classification layer, and you just use to extract embeddings of your input data. So in our case, if we have 3 images that we feed through the neural network, we obtain 3 embedding vectors, in this case of 64 features. And then the author in their paper apply a pre-processing step consisting of first doing a PCA dimensional reduction and then applying L2 normalization on the resulting PCs. To which they apply a clustering method. In their papers, they mostly focus on k-means, but really you can use any clustering method that you like. And you can use this clustering result to assign a pseudo class to each of your input. Then you can use a pseudo class as pseudo labels in a standard classification layer. And now you can start training, right? If you feed your images through the neural network once more, you will obtain probabilities of belonging to the pseudo classes on which you can compute cross entropy loss, and you can backpropagate and train the whole thing. So the whole idea behind deep cluster is pretty simple, but the framework is very powerful in my opinion because of its flexibility. So of course, it's completely agnostic to the neural network that you're using. And so we were very interested in plugging MSNet, which is specialized in analyzing high content images. But then you can also play with pre-processing, and I'll show in the next slide why this is very important for analyzing high content images. You can also change a clustering algorithm, and in our paper we tried a bunch. And of course, you can play with the way you normalize your input. So why is pre-processing so crucial? Well, it's because of so-called batch effect. So in the BBBC2021 dataset, cells are treated on 55 96 well plates across 10 batches. And what you see on the plot here is a PCA projection of untreated wells across those 10 batches. And each dot is colored by the batch it corresponds to. And you can already visually see that a lot of batches tend to cluster together. And if you look at images coming from different batches, you can also notice striking visual difference between images. You can also quantify that with a statistical test. So here we performed an ANOVA between primary components and plates. And we notice that for each of the first 8 primary components, we have very strong statistical association with batch. So the idea is to leverage the idea that all untreated wells should look similar and use this information to align them across plates. And so for that, we combine 2 state-of-the-art techniques for batch correction. One that is called combat, that use treatment information as covariates using an empirical based method, and the one that was published more recently by Google Research called typical variation normalization, or TVN, where it does a PCA to align wells of untreated wells of individual plates, and then it applies a transformation to all embeddings, to the treated wells basically. And so you can see on the left a heat map of the cosine distance among untreated wells before we do anything, and you notice a lot of structure. And after we do the alignment, the heat map is now completely flat, and the structure is gone. To visualize that also on the previous plots, if you apply combat plus TVN, then you notice that all untreated plates now are mixed together, they don't cluster by batch anymore. And if you apply an ANOVA, then you notice almost no statistical effect of batch on PCs. And so combining all these things, we came up with our method that we called UMM discovery for unsupervised multiscale mode of action discovery, where we use the deep cluster backend, but we used an adjusted MSNet where we added residual layers to Williams' method because we found it improved drastically the results. That we use for extracting embeddings, and then for our pre-processing during training we do combat TVN, then we apply a t-SNE dimensional reduction before doing an L2 normalization. We use an adjusted k-means clustering algorithm, And also on the input, we do a illumination correction, a normalization, a Z-score normalization, and we tile them. And so if we can visualize how the training procedure clusters images in the data set. So here what you see are the feature embeddings for images and that are colored by mode of action before any training. And after one epoch already, you can start to see some clusters emerge. And if you follow the plot through the training, you notice that very quickly, so here after 110 epochs, images cluster almost perfectly by mode of action. And if you visualize them, then the images belonging to different clusters also tend to look very different from each other. So now the question is how to evaluate our approach. So the first thing you can do is, which is very useful to compare yourself to other methods, is to compute the accuracy on the labeled subset of the dataset. And for that, we align with what's most common in the literature. We use the nearest neighbor classifications, where we aggregate embeddings to one feature vector per treatment, and we assign mode of action from the nearest neighbor using cosine distance in a leave one out fashion. But if you want to evaluate your approach more globally, also on unlabeled image, it's useful to use clustering quality metrics. So in our case, we use the silhouette score, which is a global clustering quality metric that compares the mean intra-cluster and mean nearest cluster distances. And higher values of the silhouette score reflect more homogeneous, better separated cluster. How well UMM discovery is doing? Well, like MSNet, we achieve 100% accuracy on 9 out of 12 of the mode of action, and we do a very good job on the remaining 3, obtaining always above 80% accuracy. And we have a total accuracy of 97%. If we compare ourselves to other methods using the nearest neighbor classification approach, we notice that we do slightly better, and most importantly, we do slightly better, but in a completely unsupervised framework, and also using a neural network that is specialized for high content images. Interestingly, we can also compare ourselves to supervised training. So here we use MSNet, trained to predict mode of actions, either without batch correction or with batch correction. And we notice that both on the nearest neighbor accuracy and on the silhouette score, we do actually pretty closely to supervised training, which is, in my opinion, quite impressive for a fully unsupervised technique to do as well as something that is actually trained to predict the labels you're interested in. But again, most interestingly is you can use an unsupervised technique to predict the mode of action of unlabeled compound. And so here on the left, you see a t-SNP plot of all the entire data set, where embeddings are colored by their mode of action, and in gray you have all the unlabeled images. And we picked 4 clusters in this dataset. And if you look at cluster A, for instance, all compounds that are annotated within cluster A are annotated as DNA replication. But you also find compounds that are not annotated in this dataset, such as bleomycin. And if you look at the drug bank target, bleomycin is annotated as a DNA ligase 1 inhibitor, fitting very well with the DNA replication mode of action. If you look at cluster B, most compounds in the cluster B are annotated as DNA damage, and this compound here is not annotated in the dataset. And again, if you look in drug bank, the target in drug bank is very well aligned with the DNA damage mode of action. Looking at cluster C, all annotated compounds in cluster C are annotated as being microtubule destabilizers, and a bunch of compounds in cluster C are not annotated, And we found 2 in drug banks that are annotated as tubulin beta chain inhibitor. So again, fitting very closely with the mode of action of this cluster. Then you can also look at clusters where you have no annotated compounds. And interestingly, we picked cluster D because we noticed that all compounds in cluster D have very different mode of action. You have some statins, you also have some cytotoxic agents that are used for the treatment of AML, and they don't seem to fit together. But if you visually inspect the images, you notice that very few cells are left on those images. And so here what we're really capturing is the anti-proliferative or cytotoxicity of these agents are at high dose. You can notice also that high doses are found in this cluster. And also what's interesting is if you follow a single compound, or in this case 2 compounds at different dose, you'll see that their cluster assignment will change drastically. So for example, for this one at a very low dose, it goes with its mode of action, and then when you increase the dose, you can follow its trajectory across the dataset before it falls into the cytotoxic mode of action, let's say, when you reach very high dose. And same for this other compound. So to conclude, UMM discovery, our method achieved very good results. That is on par with supervised learning, but in a fully unsupervised fashion. And the method can differentiate between mode of action effects and proliferation effects. Discovery of a novel mode of action is possible, but in my eyes, more importantly, it's well suited in the lead optimization phase to study time and dose course experiments, because it makes no assumptions whatsoever about the treatment that is applied to the cells. We're still working on improving the performances, and in particular, we're looking for alternatives to the clustering backend, which currently still requires tuning of several hyperparameters, and we're interested in making the method even more unsupervised. So I want to acknowledge my co-authors, and in particular, I want to give special credit to Rens, who is the lead author on this study and did most of the work that I presented today. And the paper is currently a preprint on bioRxiv, and we're in the process of submitting it to a peer-reviewed paper. Thank you for your attention, and I'm happy to take questions.